My screen is visible.
Yes, ma'am.
Okay. Okay. So in the last week or week 8, you all have studied about the fermentation of different products like glutamic acid. Then you studied about the two different antibiotics. That is, first one you have studied about penicillin, then streptomycin. So today's assignment is based on those three lectures. So let's start. All the questions are really simple. You only need to know a little, like a idea of every side of a particular product. Just like, let's start. The first question is the following chemical structure. If there is any problem in my voice and screen, then please let me know. The following chemical structure represent which of the following compound? So there are four different options they have given. First one is glutamic acid, second is 2-amino glutaric acid, third is 4-amino pentadioic acid, and the fourth option is lactic acid. So we have to tell about this chemical structure. Okay, let's start. When we like name any chemical compound using the IUPAC standard, and if there is a carbon chain in that chemical compound, then first what we have to do, we have to numbering it from a particular side, and that side you can't change that. So it depend upon on which side the chemical group main chemical group is present. From there you start the numbering. Okay, so like in this structure you will numbering it started it from here. Like this is one, here it is also carboxylic acid, this side also carboxylic acid. So from in this in this manner the both the sides are same, so then you will consider the second one. Then you will check what is present on the second option, second place. So you are seeing here that here NH2 group is present, so this is your second carbon, means this numbering will start from here. So it will be like 1 2 3 4 and 5. So for naming this compound, what you have to do, first you have to do the numbering to mark the number of carbon and to mark the position of any functional group that is not the major. Like in this, carboxylic acid is the main functional group and this is the secondary. So when you name this compound after the numbering, then how will you name this? That what are the group present at the different position? Suppose at the second carbon amino group is present. Like this is the very simple naming that you will do at the first. That is 2 amino. Then how much carbon are there? You can count it like 1 2 3 4 5. So when 5 carbon are present, what word you use? You will use pent. Then it is like, and there is no double bond in the carbons. In all the 5 carbon there are single bond present. So this that is pentane dioic, because in this both the sides carboxylic acid are present. So we'll name it 2 amino pentane dioic acid. Okay, this is the like very first name using the IUPAC when you don't know the any general name of for this compound. So this is the first name that we can use. Then for this there is like general name of this compound is glutamic acid, because this is a very common compound. So few compounds are named differently apart from IUPAC. so that they are easy to remember. So this is also glutamic acid. And the third one, you can also write it in a like 2 amino, because at the second group amino group is present, and this structure is named as glutaric acid. So this is also the name for this compound. Okay, so for this question answer is first one. Let's read the glutamic acid. Yeah, correct. Then second option is 2 amino glutaric acid. This is also correct. And the third option is 4 amino pentane dioic acid. This is wrong, because this is not 4 amino, this is 2 amino. So you have to remember the position, or you should know the like numbering procedure from where to start the numbering. Okay. And lactic acid, no. So for this question answer is A and B. Yeah. Can anyone tell me how many carbons are present in lactic acid? All of you have studied about lactic acid, so you must know the structure. Okay, lactic acid is a like 3 carbon compound. Okay, in which you have both OH and COH group. So you must know about all the options also. Now coming to second question. Okay, let's discuss a little bit of, because in the coming slides we are going to discuss about specially 2 antibiotics. That is, first one you have studied about penicillin, and the second option is streptomycin. So these two antibiotic fermentation process you have studied in the previous session. So both the antibiotics, that is penicillin and streptomycins, are secondary metabolites. What are the secondary metabolites? Like antibiotics, pigment, toxins, and others. So what happen when the growth of any microorganism occur? Then it occur usually in 4 different phases. First one is lag phase, second one is log phase, third one is a stationary phase, and the fourth one after this is like decline phase or death phase. Okay, this is usually a life cycle of any microorganism. The time period of all the different phases will vary from microorganism to microorganism, but these 4 stages will remain for every microorganism. So what are the secondary metabolites? Secondary metabolites are the compounds which usually form when the fully growth of that microorganism occur, usually in the stationary phase. So the when suppose this microorganism are growing, they start growing and they are growing at an exponential phase, then after a little bit of saturation or stationary phase come. So the compound that are released during this stationary phase are called as secondary metabolite. Okay, that usually occur means when they are in stationary phase, little bit of like they are living in a strain condition. So at that time the metabolites or the compound that are produced by those microorganisms are called as secondary metabolite, and the microorganism that usually occur at the log phase especially, means this is the growth phase, they are called as primary metabolite. Okay, so both the penicillin that we will discuss in the coming questions, penicillin and streptomycin, are secondary metabolite. Okay, you can check it from the graph that this blue curve is showing of primary metabolite, that the concentration of primary metabolite is increasing as the growth is occurring, means especially in the log phase the concentration of that is increasing. But the secondary metabolite that is shown here with a dash line, they will usually start forming at the stationary phase. Okay, so you must know the difference of these two. And what are the examples of compound that form during that are example of primary metabolites are like this: amino acids, nucleosides, vitamins, enzymes, and other products also. And different toxins also produced at the secondary metabolite. Okay, just a minute. So now coming to the next question. So what they are asking, which of the following microorganism can be used for the production of glutamic acid through biological routes? So they are asking what is the microorganism you will use for the production of this glutamic acid. Options are Micrococcus glutamicus, Strep Staphylococcus aureus, Sarcina ventriculae, and none of the above. So any one of you want to answer this? Because at least you must know this, like which microorganism is used for its production. Okay, for any fermentation, because here what we are studying the fermentation of the different products. So what are the four major things that you required? You must know about the growth factors or the growth condition required. Then what is the microorganism used, and the fermentation different conditions, and the final purification, or you can say downstream processing or purification. So you must know all those things. So this is really important microorganism. So for the production of this glutamic acid, this Micrococcus glutamicus is used. So for this question answer is A. Okay, this is the next question. The downstream processing for glutamic acid production involves a decolorizing step. Which of the following component is are used for it? Options are: first one is activated carbon, second is sodium sulfide, third is both A and B, and D option is none of the above.
Activated carbon.
Correct. Activated carbon is used, but yeah, Sadhika correct, both. This sodium sulfide is also used for this decolorization step of glutamic acid. So as we have discussed in the previous session, that decolorizing step is used in the processing, in the initial steps. So you must know the every step, and finally purification part. So for this decolorization, both activated carbon and sodium sulfide are used. So for this answer is C. Now coming to next question. Which of the following is a side effect of using glutamic acid? So you must know that glutamic acid is used in a various type of applications. So they are asking about the side effects. This is also used in foods, cosmetic industry, other industrial applications also. So these are the options, like what are the harmful or side effects of using this glutamic acid. First one is it causes sweating. Okay, second option, causes high blood pressure. Third is causes tooth decay and fattening. And D is all of the above. So using glutamic acid causes sweating. Yeah, correct. Uh, it causes high blood pressure. Yeah, that is also correct. And tooth decay and fattening. This is also correct. So all these three are the side effects of this glutamic acid. So for this question, answer is D. All of you have studied about this. That's why I'm not explaining these in like much detail, or these are only the side effects. So you must know at least a bit what are the advantages, what are the disadvantages, and what are the growth factors that is required, what is the pH, temperature, microorganism used. And then for different bioprocess, for different downstream processing steps, you must know what are the compounds used for any particular component. Suppose glutamic acid, decolorizing agent. This activated carbon is also used in previously like wine production and beer. But this sodium sulfide that we used, yeah, sodium sulfide, can this be used for this previously wine and alcohol production? That you must know this difference. And few common steps that are like applicable to all. So for this question, answer is D. This is the next question. The broth after glutamate fermentation can be hydrolyzed by. Okay. So what are the options? First one is sulfuric acid, second is hydrochloric acid, and the third option is acetic acid. Or D, all of the above. So any one of you want to answer this?
Sulfuric acid, ma'am.
Sorry, sulfuric acid.
Yeah, sulfuric acid.
Okay. No. For glutamate fermentation, we use this HCl acid. A- And why this hydrolysis, or after this is mainly the step of this downstream, and in downstream especially when you finally want to purification steps. So in that different parts or different components are used. So for this glutamate fermentation hydrolysis we use HCl. So for this question answer is B. Okay, next question is penicillin production can be improved by the following method or methods. Saptika, yeah, correct. But let's read the option first. First one is generating mutants using UV irradiation. Okay. Second one is improvement in composition of the medium. Correct. Then the third option is isolating better penicillin producing mold. And the D is all of the above. So what happened when initially at the very first time when this penicillin is produced, the amount of that is really very small, like 5 mg per decimeter cube. Then when you change the different growth conditions or growth vectors, you can change, right? Or the second factor that is majorly, that is microorganism. So when you either change this or in change in this. So when you change the growth vectors, then possibly the production will change. And when you change the microorganism, then also the amount that is produced is changed. Like this is the initial at the very first when this penicillin production start. But now when we change, and initially which penicillin is used, like which microorganism is used for producing this? Can anyone tell me the name of which penicillin is initially or first used to produce this? Or now at present which microorganism is used? Penicillin G. Now I'm asking about the name of microorganism. This you are telling like which type of penicillin is produced. I'm you asking which type of, like which microorganism is initially used for the production of this penicillin. Initially at the very first. Because at that time the amount that is produced is 5 mg per decimeter cube. But now when you change the microorganism, it will go up to 50 gram per. So it's a huge change in amount. So this occurred due to change in microorganism. Okay. Initially the penicillium notatum is used. But now which microorganism is used? That is penicillium cry. Now you will use different variety of this penicillium cryos. Sorry, I'm forgetting that name. But the different species of this penicillium is used. So that's why,
Penicillium chrysogenum, ma'am.
yeah, correct. Penicillium chrysogenum that is used nowadays. So when you change the species, the amount will change. So for our industrial production, what we require? The species that produce more and more amount of this penicillin. Okay. So from this option that iso improvement in composition of medium, that is also correct. When you change the media, the amount will change. And generating mutant using UV radiation. It means that suppose there is a microorganism A. Okay. When you use this UV radiation, suppose there is some change in the gene of this A occur, and it will start forming a different type of protein or any, like different type of any compound that is producing differently, and this will help in increasing the production. So this is also a type of like species change, but not completely species change, but there is little bit change in A species. So it will become suppose A dash, a little bit change in occurring the function of this. So this type of mutant will occur. And mutant can be done not only using UV radiation but can be other factors also. But we have to take care of that, that whether the bacteria in which you want to do this UV radiation, that is possible or not using the different methods. Mutant simply means you are changing a few factor in a species A so that it will start. Suppose, okay, I will explain it again. Suppose there is a bacteria A and it is forming a compound X only 5 units. Okay, when you when there is a mutation occur in A, it will start forming the amount of X as a like 8 units. So this is a example of mutation, that due to mutation there is some change in that microorganism and that will lead to change in amount of the required component. Okay, so using all the three methods we can change the penicillin production. So for this question answer is D. Okay, this is the next question, that penicillin belongs to which group of antibiotics? This is really simple question. Options are alpha lactam, second is beta lactam, and third one is gamma lactam. So which type of ring is present in this penicillin structure? Yeah, beta lactam. Correct. So all of you must know penicillin structure, at least the various structure present in this whole penicillin structure, that you must know. Like this is the structure of penicillin. So this is the beta lactam ring. Then this is a thiazolidine ring, and this is a side chain. So when you, this is a like general structure of penicillin, but when you change this R group, then the different types of penicillins are produced. So with the change of this, different penicillins are produced, and they can be used in they can be used differently. Okay, so for this question answer is B, that beta lactam ring is present in penicillin. Then this is the next question, which of the following penicillin group of antibiotics is derived through synthetic methods? So options are kanamycin, ampicillin, streptomycin, and carbonicillin. So what are the, first you must know like what is the difference between this synthetic method and the natural method. Actually what happened when you produce the penicillin, when you use a fermentation process and directly the penicillin that is produced using the microorganism, it is called as natural penicillin. And thus in synthetic method what you will do, you obtain the penicillin but you did the different chemical changes or any different structural changes or any chemical you can use. So there is a change in structure of that. So the so those penicillin that are produced after this treatment, that are called as synthetic, and that we use directly from the produced from the microorganism, that are called as natural. Okay, so from this which are the synthetic one? So from these four options, this ampicillin and carbonicillin, these two, for this answer is B and D, that ampicillin and carbonicillin are used as a synthetic method, are formed using the synthetic method. Okay, this is the next question, that penicillin is effective against the following microorganism. First option is gram positive bacteria, second is gram negative bacteria, third is all bacteria and fungi, and the fourth is fungi. So this penicillin is a antibiotic. So from this you must know that it will work against bacteria. Okay, so these two options are not correct. It won't work against the fungus. So then you must know like which bacteria, either gram positive or gram negative or both. So how do you know this? And for every antibiotic, like for those you will study in detail, you must know the pathway, like how this penicillin or any other antibiotic or any component, how it will work. So for this you must know the pathway of this penicillin. It usually work on gram positive bacteria. Okay, let's discuss how. we will this is the membrane structure of gram positive and gram negative, that you must remember that in the gram positive a outer peptidoglycan layer, a thick peptidoglycan layer is present. Okay, but in the gram negative bacteria only a single or like very thin layer of peptidoglycan is present. So what happened, or what this penicillin did, it usually attack on this peptidoglycan layer. So in this gram negative bacteria, as you can see, there is very thin layer and it won't be like if be able to destroy it properly, and because there is outer layer also from this peptide, like after this peptidoglycan there is a outer layer also, so the penicillin is not able to like move inside the bacteria, these gram negative bacteria. But when gram positive bacteria is present, it usually like it won't allow the formation of this peptidoglycan layer, or it will enter the bacteria. So when it will enter, it will, due to osmotic shock, this bacteria will destroy or die, because it won't allow the formation of this layer, this peptidoglycan layer. Okay, like see, this is the bacteria and this is the outer layer they are showing. This is a gram positive bacteria, gram negative bacteria they are showing. So in this there is only a single like periplasma, or you can that I have shown in here that peptidoglycan layer is present, but here the full layer of this peptidoglycan is present. So this peptidoglycan is usually made up of these two components, N-acetyl muramic acid and N-acetyl glucosamine or glutamic acid, you can say. So these two components, that NAM and NAG, that we usually call in a simpler way. So there is a bonding between these two for the formation of this peptidoglycan layer. So what penicillin did, it won't allow the formation or the merging of these two components. So when this will not occur, the peptidoglycan layer will not form. When this will not form, gram positive bacteria will not survive. Okay, so understood like how it will work between the gram negative and gram positive bacteria, and why this will not work against micro like humans, because in our cells what happened, cell wall is not present, so it won't attack human cell, it will only attack the bacterium cell, and which bacteria is specifically gram positive. Okay, so for this question answer is A. This is the next question, like which of the following salt can be used to precipitate penicillin. Okay, like this is only to learn like which salt is used finally to precipitate, and after this precipitate step what will occur, final purification. Okay, so which salt is used to precipitate the penicillin? Options are sodium, potassium, calcium, or both sodium and potassium. So for this precipitation, potassium is used. Okay, so answer to this question is B. Coming to the next question. Okay, match column A with its function mentioned in column B with respect to penicillin fermentation. Means they are giving some components that are used in penicillin fermentation. So we must know what are the functions of those different compounds. Let's read it. First one is calcium. Second is phenyl acetic acid or phenoxyacetic acid. Third is butyl acetate, and fourth option is charcoal. In column B, options are elimination of pyrogens, second is liquid extraction, third is precursors, and the D is counter acidity of corn steep liquor. Okay, for the penicillin fermentation, what is the precursor? What does this precursor mean? Means the initial component that is required for its production. So for the penicillin production, this phenylacetic acid, or you can call it phenoxyacetic acid, this is used as a precursor, means this is the initial component that is required. So for this, second is C. So in the two options they have given that second, they have matched second with C. Okay, then we must know like function of charcoal. We can say charcoal is mainly used for elimination, or as we discussed previously, that decoloration. So it means it is mainly used for removing or elimination of components. So charcoal is used for elimination of pyrogens. So for this, fourth is matched with A. So now both the options, till now both the options are, both A and C are possible. So then we will see about calcium. So why calcium is used? Calcium is mainly used this for this counter acidity of corn strip liquor, because it will precipitate if we use the salt of calcium, and it won't allow the change in pH. So this calcium is used to counter acidity of this, and this butyl acetate is used for the liquid extraction. So for this question answer is A. Okay, means you must know that functions of all the components that are used, or at least the major components that are used, because only that when you know the difference, you can differentiate between the production of two. Like here you are discussing about penicillin, then similarly if you ask about the streptomycin, so you will be able to differentiate when you know the functions of the major components that you will use. Coming to next question. This is match column A with column B in terms of pH maintained during penicillin fermentation. So they have given 3 pH, that pH 2 to 2.5, pH 7.5, pH 8, and here three options are given, that extraction in aqueous phase, extraction in solvent phase, and the third way is production stop. So what happened in the penicillin fermentation, when you increase the pH like beyond this, or beyond like around 8, or this penicillin production will stop, means it won't form at a very basic pH. So you must know at least which pH, till which pH it will form or not form. So when you know this, then you will know at least this, that the pH, when pH is very basic like 8, the production of this will stop. So for this, first one is meshed with C. Okay, then this pH 2 to 2.5, this is the pH that is initially present. So this is the extraction in solvent phase, and when it is finally in a aqueous phase, this pH 7.5 will work. Okay, so for all the three different pH, we have different part, or different you can say activity of this penicillin. So for this question answer is A, that pH 8, when it will go to pH 8, the production of the penicillin will stop, and this pH 2 to 2.5, that is initially in a solvent phase, and pH 7.5, you can little bit get an idea of when this is a aqueous phase, then the pH will remain like close to neutral pH, not exactly neutral. So this extraction in aqueous phase will occur at pH 7.5. So for this question answer is A. Okay, this is the next question. The following drug is effective for treating disease caused by Mycobacterium avium complex. So options are amoxicillin, streptomycin, ampicillin, or all of the above? So you must answer this. Yeah, Satvika, correct. That streptomycin, because this is the first antibiotic that is used for the diseases that are occurring due to mycobacterium species. All the For the TB also, this streptomycin is the first antibiotic that is used. So for this question, answer is B. Next is, what is the mode of action of streptomycin in fighting against infection? So again, similar type of question, they are asking about the mode of action. So this mode of action is really important when you are studying about any, like antibiotic, antifungal, any type of this pharmaceutical product. Then you must know the mode of action and the major, like how it will target and whom it will target. And one major thing, like how to differentiate whether it will attack only the microorganism or it will start affecting the human cell, or why it won't attack the human cell, why it will attack only the microorganism. This difference is really important that you must know. So what are the options given? Let's read it. First is, it inhibits protein synthesis. Second is, it destroys the cell wall structure of bacteria. Third is both A and B, and D is none of the above. So this, it destroys the cell wall structure of bacteria. This is the method of penicillin. But it might be of streptomycin. So we can't say that if it is of penicillin mode of action, it can't be of streptomycin. Okay. So you must know how this streptomycin will attack. What this streptomycin will do is, this streptomycin will stop the formation of like translation step. It will, okay, I will show you here. It stop the formation of proteins. Okay, this is the, see, this is the ribosome structure. Ribosome mainly contain two units, the small and the larger unit. So this is the 30s unit. So what streptomycin will do, it will stop the functioning of this small unit, 30s subunit. So when the function of this will stop, then from the mRNA to this protein translation won't occur. So when there is no protein, mRNA to protein. So this step will not occur. So when there is no protein formation, the microorganism will not survive. It will die. Okay. So it will stop this procedure, and this step is called as translation. Like this is the general step of formation of any proteins in our, in any microorganism, that from DNA to mRNA, mRNA to protein. So this is the translation step, and this is transcription step. Okay, so it will stop this process. So when protein won't form, the microorganism will die. Then how it will differentiate between, like if this antibiotic only won't have to attack the microorganism, not the human cell, because this 30s subunit of the microorganism or bacteria is different from our small unit. So that's why it will differentiate, it won't attack the human cell. Okay, so for this question, Satvika, both A and B. No, it won't attack the cell wall structure, Satvika. It will attack the, like it stops the protein synthesis, and when there is no protein formation, the microorganism will not survive. The cell wall structure of bacteria will be destroyed by the penicillin. So for this question, answer is A. Now coming to next question. Okay, this is very simple question. That streptomycin fermentation is a what? Aerobic, anaerobic, can shift between aerobic and anaerobic, or all of the above. So anyone want to answer this? Like how this fermentation process occur in the presence or absence of oxygen? Okay, this streptomycin is formed in the presence of oxygen, so it's a aerobic process. Okay, so for this question, answer is B. Or you can say it require oxygen for its production. Okay, this is the next question. The following statement for streptomycin production during phase 2 is are true. First is ammonia is consumed. Second option they have given glucose uptake is low. Third is weight of mycelium remain constant. And D option is all of the above. So what happened in the streptomycin production? This ammonia is consumed. That is correct. Glucose uptake is low. No, this occur when glucose uptake is low. The growth will hamper of this streptomycin. Then weight of mycelium remain constant. Correct. This is not correct because if this will hamper the growth of this streptomycin. So for this production, this is required. So for this answer is A and C. Okay, or you can call you must know that precursor of streptomycin is glucose. Like it require as a glucose or sucrose or any type of like sugar source. So for this question, answer is A and C. This is the next question. Which of the following salt is used to precipitate streptomycin? Okay, as we discussed previously about the penicillin, similarly we are studying about the streptomycin. Like we are studying on the same pattern. Like what is the mode of action, what are the salts you used to precipitate, what are the different components that are used in fermentation, and what is the function of those components. So and finally purification process. So these major steps will remain same, but like suppose this is penicillin and this is streptomycin. If we write it like mode of action, so you have to study mode of action for both, but these are different. So you must study the few major things about all the products that you will study. Okay, so coming to this question, which salt is used to precipitate a streptomycin? Option are phosphate, nitrate, sulfate, and all of the above. Sulfate. Yeah, correct, Satvika. For this, sulfate is used. And this is the last question of today's assignment. And this is really simple question. So all of you must know that baker's yeast can convert fermentable sugar present in bread dough into what component? That carbon dioxide and methanol, carbon monoxide and ethanol, and carbon dioxide and ethanol. Okay. Option C. Anyone want to say any other option? Okay, this is correct that when this baker yeast is used to convert this fermentable sugar convert this sugar into component, then CO2 and ethanol is formed. Or you can write it like this is a very C6H12O6, like this is a very famous or very common equation. You can call it C2H5OH. Formed, and you must balance this equation. Okay. So when you ferment this fermentable sugar, it will form CO2 and ethanol. Okay. For this answer is C. So these are the questions related to your week 8 assignment. So if anyone of you have any doubt, please let me know, or any suggestion you want to give, then also. Okay, if there is no doubt, then I'll stop the recording.